Good afternoon, everyone.
Hello, please, am I allowed to make
I
a
also look
question?
forward to the paper. I am Dr. Akpoka, who is Augustine. I'm an immunologist, a cytokinologist, a nurse scientist, a microbiologist, and an immunomycist. And I am collaborating on this paper with 5 others. Dr. Aigbe DLA, a criminologist, Izevbuwa OE, who is a forensic scientist, a medical laboratory scientist, a molecular virologist, and a microbiologist. Ashaka OS, who is a virologist and a molecular virologist, and Ochem CA, who is a professor of law. And so we wanted to look at it in total. And that is what we are going to do here today. A number of us have never heard of the term forensic immunology. And that is because we often, as already mentioned by previous speakers, narrow our mind regarding forensics. I was just telling my colleague out there just now that I am not happy that we don't have microbiologists here. I am not happy we don't have biochemists here. I'm not happy we don't have nursing students here, because we have nurses. I'm not talking about staff, I'm talking about students. I knew of this conference 2 days ago, and I told them I must be part of it, just 2 days ago. We have nurse forensics. Nursing students can major in forensics. Microbiologists can major in forensics. Immunologists like myself can major in forensics or consult in that area, and that is why we have immunological forensics or forensics immunology. And then we have Panomics. Panomics has to do with what they call multi-omics. Okay, multi-omics, you have areas as genomics, transcriptomics, that's transcription. For those, the molecular people talk about DNA and all of that. Then you have proteomics, talking about protein, metabolomics, and epigenomics. So we can actually, or it is already done, combine these omics, known as multi or multi-omics, to solve crime. And we are going to see certain examples and global best practices where these are already being employed. We know very well that forensic science has evolved, especially with technological advancements. And it has now moved beyond the traditional fingerprinting that we know, blood typing and serology, toward molecular techniques that offer higher precision and reliability. KSAT 2021. The emergence of Panomics, I mean integrating all the omics, has now opened new possibilities for forensic analysis. Now, one of the challenges that forensic scientists usually have, or forensic operators usually have, is determining the cause of death, because it's a fundamental aspect of forensic science, and it usually involves several investigation techniques to distinguish between natural death and unnatural deaths. For instance, in Nigeria, if you see two people fighting and probably as a result of the struggle, one falls down and dies, the other person will be held liable. But in other parts of the world, that's not the case. You have to determine how, why the person died, to be able to say that the person committed either manslaughter, murder, or The couple homicide can also be murder, depends on the code that is being used. All right. Now, when we talk about microbial forensics, it is an emerging field. We, instead of looking at the DNA, we use the microbial. Okay, I'll explain a little of these terms because we are not, most of us are not scientists. We have what we call microbial signature. Every person here, there are microbial signature in your palm, your nose, all parts of your body. And so it means that I can take samples from you and do a typing to understand the microbial signature that will tell a story. It will tell us where you have been, where you have been, the people you have interacted with, and all of that. And it has wide-ranging applications, even in contact tracing during epidemics, pandemics, and what have you. And so microbial forensics use that foundation, to investigate crime and solve other problems. I'm trying to rush through the general so that I can bring out cases, where this has been used. The same thing with immunological forensics or forensics immunology. There are biomarkers in the body that can tell a story. For instance, time of death. Is the death as a result of violent confrontation or was it natural? There are cases where mothers have been charged for murder. Mothers have been charged for murder in infant deaths, sudden infant death. There's something we call in medicine, there's something we call sudden infant death. And some of these sudden infant deaths may not even have a known cause. Somebody just kept a baby here, you go out and return and the baby dies. It is even easier on the mothers. What of nannies? You leave a child for a nanny and then you return to see that the nanny, the baby is dead. And so that becomes a problem. And so there are immunological markers, which I'm going to look at, that can tell if that baby died of natural causes or if something happened that shouldn't have happened. And also, let's go down to forensic immunology and crime investigations. I already mentioned immune biomarkers, and one of them is the one that we use in cause of death. We have what we call cytokines. Remember I told you that cytokinology. It means that I study cytokines. Cytokines are proteins, they are living proteins, and that is one of the ways that the immune system interacts with each other. And there are several of them. One of them that is mostly used by forensic immunologists is what they call the interleukin 6 and the tumor necrosis factor and the interleukin 10. Those are biomarkers. Now, these three biomarkers I mentioned, they are inflammatory mediators, inflammation, okay, and they can provide insight into immune responses during infections, trauma, and systemic inflammatory reactions. And so, you can tell that is this death as a result of infection, is it as a result of trauma, or other things. Then we have another one, they call it the C-reactive proteins. We have C-reactive protein, CRP, and we have what they call the serum amyloid, SAA. In acute phase proteins, C-reactive proteins and SSA are markers of inflammation and have been found to be elevated in pneumonia-related death, reinforcing their role in forensic differentiation. For instance, and as a result of that, one person hit the other one's neck. Okay, and then the person dies. The person in this line will be charged for murder, okay, or mass murder. But in other crimes, they can check and say, what actually happened? Was this altercation enough to cause the death of this person? And in that case, you can look at some of those biomarkers. We also have tryptase. Tryptase is produced by the mast cell, they are immune cells, and they are key biomarkers in anaphylaxis. Anaphylaxis are allergies. Okay, they are allergies which can be severe or threatening. They are severe allergic reactions when you get one allergy. It can happen immediately or after a few minutes. We can also refer to it as allergic shock or anaphylactic shock. Okay, when you use the word shock in medicine, we are referring to the tissues not able to get enough blood and so enough oxygen and all of that. So enough oxygen. We have a lot of acid shock, anaphylactic shock, hypovolemic shock, and all these kind of shocks. But in all the cases of shocks, they are saying that the tissue is not able to get enough oxygen. And so that can happen. And of course, we know that if you do anatomy and physiology, you understand that the time that it takes when somebody is starved of oxygen to die is a very short time. And even when they survive, the person may not be able to function well again. So it's very, very important and important. We also have these shock proteins, the HSP70. They are also used, especially in stress management and looking at death through heat strokes. Heat strokes, you know, in places where you have heat. Of course, we have already experienced a lot of this now. And animals and even some people in certain areas are victims. I saved 2 dogs yesterday, 2 dogs who almost died of heat strokes. I went somewhere to do some business and I ran into them. They were already choking. I had to start pouring them water and that, but they were revived. 2 dogs. All right, so we have IgM and IgG. Without going into the details, they are called immunoglobulins. Immunoglobulins are produced by your white blood cells, a subset of your white blood cells. We call them leukocytes. Okay, and the leukocyte is your white blood cells. We call them lymphocytes. Sorry, we have the B lymphocyte and the T lymphocyte. The B lymphocyte produces immunoglobulins. And those immunoglobulins are about 5 classes. We have IgG, IgM, IgA, IgE, and IgD. They are called antibodies. Antibodies is another name for immunoglobulins. Those immunoglobulins, especially IgM and IgG, can tell whether somebody had previous infection or immediate infection, and they have long found usefulness in forensics determination. Especially in the case of Epstein-Barr virus, there's a disease they call the kissing disease. The kissing disease, the big name for it is mononucleosis, and there are complications that can come as a result of that. And forensics, especially immunological forensics, has been used in that case. Now, so there are several explanations. I already mentioned that of the sudden infant death, and several others. For instance, to establish drowning, there is a protein that can be used. We call it the SPS. We call it the SPS protein, that is surfactant proteins. There are 3 classes of surfactant proteins. We have the SPA, SPP, and SPD. But by determining that in case of drowning, we are mostly interested in the SPA and the SPD. The SPP is the reason your lungs is able to expand. But the SPA and the SPD also play certain, they are useful in that determination. Now, let's look at notable global cases where this has been employed. We have the Lucilette case that concluded in 2025 and recently has been opened. The Lucilette B was a nurse. She was a neonatal nurse. Okay, that means a nurse that took care of children, little children. And then they realized that there were several unexplained deaths within the ward she was working whenever she was on duty. And so they started investigating, and what actually the last one they coffee was immunology, where they were able to look at abnormal insulin in those victims that died. Of course, if you go back to look at this case, you realize that it has begun to generate new attention because of the complexity because of the method they utilize in reaching the conviction. Those are still in jail, but the case has just been revisited. Then we have COVID-19 post-mortem investigation. When you look at data, especially World Health Organization data, you'll be told that some number of people died as a result of COVID-19. It was those numbers that were used as against other deaths. You have more than 16 cases of 1994 in the US. You have the presidential president in Russia. We have the mycotoxin poison outbreak in Kenya, that was in 2004, and several others. Now, we have challenges in Nigeria's forensic immunology because this paper will not be complete without situating it within the Nigerian context. And other speakers already said that we are having limited forensic infrastructure. I am not too sure that we have more than one forensic infrastructure in Nigeria today. If you are going to carry out those investigations, you will have to take it from one lab to the other. You may not have a one-stop shop lab that will be able to attend to all the needs as regards forensics. We have shortages of skilled professionals in the field. The good thing is that one of the co-collaborators in this project is one of the top forensic scientists in Nigeria today. I should introduce you to the association to be a member. I'm talking about Dr. Izevbuwa. He's a rising forensic expert. Then we have legal policy barriers. For instance, the Evidence Act of 2011, which I heard mentioned earlier, does not explicitly recognize forensic immunology as admissible evidence, and so that also limits use in legal proceedings. It means that if you go all out and come back, they'll tell you the existing law does not support this evidence, and it will be thrown out. And so there are recommendations for that. We have establishment of forensic immunology lab, legislative reforms, capacity building, and international collaborations. I will watch out for the community that would emanate from this conference to see how we approach these things. And there are several incidences where microbial forensics were used in bioterrorism. The commonest bioterrorism we know is the anthrax caused by the Microbial anthracis, that can be fatal to man and animals. And several cases, for instance, a case of the United States in 2009, it was as a result of microbial forensics they were able to trace it to the lab to realize that the person that released it to the public was a military scientist. So they look at the signature that I mentioned earlier. We have salmonella. Salmonella is the organism that causes typhoid fever, salmonellosis, or the one you call enteric fever. It's not as common as you are made to believe in the hospital. Then you have SARS-CoV-2 origin. There are a number of persons, or there's a school of thought that believes that the SARS-CoV-2 is the virus that causes COVID-19, and other strains of it. And they believe that it was engineered. It was as a result of that genetic typing that they were able to get what they call the evolutionary relationship to know that the one that was out there in the population, although released from the lab in Wuhan, was not bioengineered. It came from the bats. So most of all those things, and of course the challenges always remain the same. Now let me mention the legal framework. I already mentioned the Evidence Act of 2011 as inadequate. The Criminal Act Code provides legal basis for prosecuting bioterrorism and contamination. Then we have the National Biosafety Management Agency. It regulates biological research. We can impart forensic immunology or forensic application. There are recommendations, and they are similar. Established microbial forensic laboratories, develop legal framework, build capacity, and international collaborations. Now, let me quickly conclude by looking at global best practice. If you look at global best practice, you realize that, okay, I've been told that I have less than 5 minutes. Okay, less than 5 minutes. So I'm going to look at certain practices globally. Global best practice in forensic panomics. I've talked a little about immunology. I've talked about the microbial aspect of it. I'm now looking at the omics aspect of it. That is the multi-omics, also known as the panomics. If we have investment in advanced forensic infrastructure, and we have a case study. I have a lot of case study in my work, and one of the case studies is FBI CODIS. FBI CODIS is the FBI has incorporated forensic genomics and microbial analysis into its crime investigation. And the CODIS was developed by FBI and is the most advanced forensic DNA database globally. Imagine, the owner of the most advanced genomic database globally is a crime or security organization. Do you understand? Not a research organization. And so we can adopt something like that in Nigeria. We also have the UK Forensic Science Regulator Act, and I think that could also be, looked at in Nigeria. By the way, this paper will be published in the journal, and most of the things most people have the opportunity, to take a look at it. And so, in conclusion, forensic immunology, microbial forensics, and panomics offer new avenues for crime scene analysis, evidence identification, and suspect tracking. While these technologies have been successfully implemented in countries like the United States and the United Kingdom, Nigeria faces due to inadequate infrastructure, legal gaps, and limited funding. Addressing these challenges through legal reforms, investment in forensic facilities, and international collaboration can modernize Nigeria's forensic science landscape and improve criminal justice outcomes. Learning from global best practices and addressing these challenges is essential for Nigeria to fully integrate forensic immunology, microbial forensics, and panomics into the Nigerian criminal system. Thank you for listening.